Okay, last time we were talking about meiosis, and we made a comparison between the mitosis and the meiosis. And now we will talk again a little bit about the consequences of the meiosis. Now, as we have seen last session, during the meiosis, we have a reduction of the chromosome number. So the meiosis allow us to have a reduction in the chromosome number. So it maintains haploidy. And this reduction of the chromosome number, this haploidy generated by the meiosis, is essential for the maintenance of the correct number of chromosomes, which is called the euploidy. So it is important for the maintenance of euploidy in the zygote and the offsprings, because when we have haploid gametes following the meiosis, this means when two haploid gametes will fuse during the fertilization, this will restore the diploid condition, the diploid number of chromosome, or the diploid situation. So in this way, we will have a diploid zygote and a maintenance of the correct number of chromosomes. If we don't have haploidy in the gamete, if the gametes are not haploids, so after fertilization, we will have cells or zygotes with more than 2 sets of chromosomes, and so which will be lethal, or it will induce damages or lethality in the zygotes. Now, as a second consequence of the meiosis, yes. As a second consequence of the meiosis, it provides a genetic diversity of individuals, as we have explained, and this genetic diversity is provided by the interchromosomal and the intrachromosomal shuffling that we have talked about, and we will talk about now in the coming slide. So the meiosis provides a genetic diversity of the individuals, and also it allows genetic sexual dimorphism. What do we mean by genetic sexual dimorphism? Do you know?
يعني في X و Y.
Yes. When we talk about sexual dimorphism, it means females and male. And when we talk about chromosomal genetic sexual dimorphism, we mean that in some gametes we will have an X chromosome, and in other gametes we will have a Y chromosome. And as you know, when we have a Y chromosome in a gamete, and this gamete will fuse with another gamete, this will be determinant to have a male phenotype, or it will, it's enough to have one Y chromosome to induce the gonads differentiation into testicles, and then to have a male. So due to the meiosis, and due to the separation of the homologous chromosomes during the anaphase 1, we can have separation of X and Y chromosomes into 2 different cells. And so some gametes will have an X chromosomes and other Y chromosomes, which will lead to sexual dimorphism later. Okay? Now, here we talked about the genetic diversity of individuals due to the meiosis. What are the mechanisms responsible for this genetic diversity? We have seen them earlier during last session, but we will review them here quickly. We have 3 mechanisms. The first one is the interchromosomal shuffling, or what we have called the independent assortment. That take place during the anaphase 1. So this is the independent separation of the chromosomes during, or assortment of the chromosomes during the anaphase 1 between the 2 poles of the cells, if you remember. Okay, so if you remember, we talked about a cell containing 24 chromosomes, so assembled in 2 pairs, each homologous pairs together. a paternal and maternal chromosomes together of a homologous pair, and then those maternal and paternal will be separated to each pole of the cell in an arbitrary way. There is no special preference to which chromosome will go to the upper pole and which one to the lower pole. And then this will make that we have an independent assortment of the chromosomes, and this will create diversity in the daughter cells. Here we can see an example. Let's take a cell containing only two sets of chromosomes and two pairs of chromosomes. Okay, this is one pair of homologous chromosomes, and this is another pair. One of them is a paternal chromosome, another is maternal, and here also. So during the during the anaphase 1, those chromosomes, the homologous one, will be separated differently to each pole. We can have either the red, let's say the paternal one, which go to the upper pole with the dark green one, and the orange with the light green to the lower pole. And in this case, we will have this cell and this cell as daughter cells. And we have another possibility where this orange one can go to the upper pole with this one, with the dark green one, and this orange, and then the red with the light green will go to the lower pole, and this will create two daughter cells, which are those ones, different from the first one, which we generated first. So here we have four possible combinations. This make so when we have two pairs of chromosomes, we can have four possible combinations of four possible gametes and four possible combinations of the chromosomes. And so we have 2 to the power 2 to the number of pairs of chromosomes possible combination. When we have 3 pairs of chromosomes, we will have 2 to the power 3, which means 8 possible combinations of the chromosomes, and then 8 different gametes, 8 gametes, which will be different from each other. Okay, so if we have 24 pairs of chromosomes, we will have 2 to the power 24 possibilities and 2 to the power 24 possible gametes. So this will create a great diversity in the gametes. Okay, this is the inter-chromosomal shuffling. So we'll have gametes with unique combination of chromosomes. Now, we talked also about the intra-chromosomal shuffling, which is the crossing over that happens during the prophase 1 of the meiosis. And during the crossing over, we said we have an exchange of allele. We said that the two chromosomes in the parental cell, let's say here, this is a pair of homologous chromosomes, they will get connected to each other, and they will get synapsed, if you remember, and we will have chiasma between them, and so physical connection between them. And at the points where we have those physical connection, we will have an exchange of genetic material. We will have an exchange of alleles between the non-sister chromatids of the homologous chromosomes, of the homologous pair. Okay, so here, for example, this is, let's say, a paternal and a maternal chromosome. We had an exchange of genetic material, of alleles. This blue chromatid of the paternal chromosome took a red, took a fragment or an allele from the maternal one, and it's the same for the maternal chromosome, which has taken a blue fragment or an allele from the paternal chromatid. Okay, so this is allele exchange, and due to this exchange of alleles, when we will have daughter cells carrying chromosomes which are not exactly the same as the parental cells. Okay, those chromosomes after the crossing over, they are recombinant. We say they are recombinant chromosomes or recombinant chromatid. Recombinant means they acquired a new genetic material. Okay, so they are not exactly the same as the initial chromosomes. They are different. Okay, so this is due to the crossing over. So as a consequence of the crossing over or the intrachromosomal shuffling, we will obtain recombinant chromatids or chromosomes, which are different from the original ones. And this will create a variety or a diversity in the gametes compared to the parent cells, because the chromosomes in the gametes will not be completely identical to the parent cells. So due to this intrachromosomal shuffling and to the interchromosomal shuffling, we will have gametes with great genetic diversity and which are different from the parent cells. And also the diversity in the offsprings can be increased due to the random selection of those gametes for the fertilization. Okay, so we have a great diversity of gametes, and those gametes are randomly selected for the fertilization. So this random selection will create also a big diversity in the offsprings. And so each zygote will represent a unique genetic assortment out of billion possible out of billion possible assortments. Okay, so those are the consequences of the meiosis. Now we will move to the second part of this chapter to talk about the gametogenesis. As we have said, the gametogenesis is the process of production of gametes. The gametes are either spermatozoa for the male and oocytes or ootides for the female, the eggs. Okay. For the reproduction to take place, we need a fusion between the two gametes, the spermatozoa and the oocytes. Okay, and the gametogenesis takes place in the gonads, which are the testicles in the case of the male and the ovaries in the case of the females. The gametogenesis in the males is called the spermatogenesis because we are producing sperm cells. And in the case of the females, it is called oogenesis because we are producing oocytes and ootides and ova or ovum. Okay, so it is called oogenesis in the females, and it occurs in the ovaries. In the males, it occurs in the testicles, which are the male gonads. Now, the gametes between both sexes, so the spermatozoa and the ovum or the oocyte, are not the same. Okay, they are. They have many. We have many differences between them, and we can mostly notice the size that is different. We can see how big is the oocyte compared to the size of a spermatozoon. Okay, one spermatozoon or sperm cell is small, very small compared to the oocyte, which is larger, much larger in size. And the oocyte is large in size because it has a large cytoplasm, a very large cytoplasm rich in reserves, as we have said, in proteins and messenger RNAs that were synthesized during the diprotein stage of the prophase 1. And those reserves are essential for the later stages of the oocyte development, for the fertilization and the first stages of the zygote divisions. Okay. However, the sperm cells, they have here a head and a tail, and the cytoplasm is present in the head region and is very small, so is reduced. It is a reduced cytoplasm, as we are going to see later. okay? And also one of the main differences is that the sperm cells are motile, however, the oocyte is not motile, okay? And in both sexes, the gametogenesis comprises 3 steps. So we have 3 common steps between both sexes, or between the spermatogenesis and the oogenesis. And those common steps are the proliferation phase, a growth phase, and a maturation phase. Now we are going to talk about each one alone. For the we will talk about the spermatogenesis. As we have said, it is the process of gametogenesis in the males, and it takes place in the testicles, and more specifically in the wall of the seminiferous tubules of the testicles. Okay, so this is a testicle, and here we have a longitudinal section of the testicles. Inside of them, we can see those tubules here. Those are the seminiferous tubules. Inside of those tubules, and more specifically in the wall of those seminiferous tubules, we will have the spermatogenesis that will occur. Okay, so those are seminiferous tubules. If we take one tubule and make a cross section of it, okay, to see what is inside of a tubule, we can see this section here. So this is a cross section of a seminiferous tubule. We can see here we have the wall of the tubule, or its edge, the edge or the wall of the seminiferous tubule, and inside here is the lumen. So this is the empty space inside of the tube or of the tubule. Now, as you can see here, we have those dots near to the wall of the tubules, and those are the nuclei of the different cells. Okay, so those are the cells at the origin of spermatogenesis. Okay, which are near to the wall of the seminiferous tubules. We will have the spermatogonia, the primary spermatocytes, the secondary spermatocytes, the spermatid, and then the sperm cells, all of them here. And we will find the most immature cells will be adjacent, directly adjacent to the wall. And more the cells will get mature or differentiated, more we will go in the direction of the lumen. So the spermatogonia, which are the most immature cells, we'll find them here next to the wall of the tube, of the seminiferous tube. And then we will have the spermatocytes 1 more inside, and then the spermatocytes 2 more also inside and closer to the lumen, and then the spermatids, and then the sperm cells, which are near to the lumen, and they will be released at the end of their differentiation. Those sperm cells will be released in the lumen of the seminiferous tubule. Okay, so the spermatogenesis begins on the outside of the tubule and progress to the inside in the direction of the lumen, okay, and progress to the inside to release the mature spermatozoa in the lumen of the tubule. And also the spermatogenesis, it begins after puberty, okay? So in the during the embryogenesis, we have a production of a pool of stem cells, of germ stem cells, and spermatogonial stem cells, and those stem cells will be maintained as stem cells, will divide and will be maintained as stem cells until puberty. Okay, so after birth, there will be nothing, there will be no spermatogenesis in the childhood. It's only after puberty that those stem cells will be activated to engage in the production of sperm cells. Okay, so the spermatogenesis begins only after puberty. Before puberty and before birth, we don't have spermatogenesis. And the spermatogenesis is continuous throughout life. This means that it does not get arrested from puberty until senescence. It is continuous. It does not get arrested. As we are going to see, for example, the meiosis in the germ cells will not be arrested during during when it is happening, and also we always have cells engaged in spermatogenesis, and this spermatogenesis will be continuous throughout the life of a male or until senescence, which means until late ages, advanced ages, okay, but will not arrest at the menopause like in the females, okay? So it is continuous during a life of a male until advanced age or until all his life, and will not be arrested at menopause like in the females. Is it clear till now?
Yes.
Yes.
Yes. Now, here we will see the different phases of the spermatogenesis. We said we have a proliferation phase, a growth phase, and a maturation phase, which are common with oogenesis. However, the differentiation phase, here we will see we have a differentiation phase that is specific for the spermatogenesis. Now, what happens during the spermatogenesis? As I have said, during the fetal life or during embryogenesis, we have primordial germ cells, which are the most stem cells, the most immature cells at the origin of the gametes, or which will give the spermatogonia and the later more mature germ cells. Okay. So during the embryogenesis, we will have production of primordial germ cells, which will divide by mitosis and to give spermatogonial stem cells, so other stem cells, but which are a little bit more mature than those primordial germ cells. And those spermatogonial stem cells will divide also by mitosis. Once they will divide, they will, as you know, each cell will divide to give two daughter cells. So those cells will divide to give an identical cell to an identical cell of the initial one. So in this way, we will keep a pool of spermatogonial stem cells, and another cell which is a little bit different, and it will become a little bit more mature. So those are the spermatogonia. Okay, so we have the primordial germ cells, which will divide by mitosis to give spermatogonial stem cells, and those spermatogonial stem cells also will divide to give identical cells and spermatogonia, which are also stem cells, okay, but a little bit more mature than the spermatogonial stem cells. So before birth, we have those spermatogonial stem cells and the production of spermatogonia, but we don't have the other steps of the spermatogenesis, okay? After birth and in the childhood, we don't have also spermatogenesis, and it's only after puberty that those spermatogonia can resume their mitotic divisions, can start again to divide and to give, so each spermatogonium, we say in singular a spermatogonium, and in the plural they are called spermatogonia. Okay, so each spermatogonium will divide to give primary spermatocytes, they can give, at certain moment, they give an identical cell. And so we will have, I will not go in the details now, we'll see them later. So the spermatogonia will divide to give primary spermatocytes. Okay, those primary spermatocytes will enter the meiosis 1 to give secondary spermatocytes, which are haploid cells, and the secondary spermatocytes will enter the meiosis 2 to give the spermatids, which are also haploid cells and having one chromatid for each chromosome. So the results of the meiosis from a primary spermatocyte is one primary spermatocyte will be at the origin of 4 spermatids, and those spermatids will differentiate later. So the spermatids will not divide by mitosis, and they will not undergo any meiosis to give the sperm cells. They will only, we say they will differentiate, which means they will have cytoplasmic modifications. Okay, they will undergo cytoplasmic modifications to become sperm cells. Now, I will talk about each phase alone. So the first step during the proliferation phase, we have the first steps of Spermatogonia proliferation, which occurs during the fetal development, then is arrested until puberty. So before puberty, we have the divisions of those cells by mitosis. Then this mitosis is arrested, and it will start again after puberty. Okay. At puberty, the spermatogonia resume their mitotic proliferation until death. So they can keep on dividing until senescence or until death. Okay, they will divide to give, to generate the initial pool of spermatogonia and to give more mature spermatogonia also and primary spermatocytes. Now, after several, yes.
بس لو سمحتي يعني نحن after before puberty يعني هو بيكون موجود نحن عندنا السبرماتوجونيوم من بعد البيبرتي بصير يطلع عندنا البرايمري سبرماتوسايت
Yes.
والكومبليت.
Yes. So here, for example, we have the spermatogonia. As you can see, they will divide. They will generate. We can have. They will divide to give an identical cell to keep on conserving this pool of stem cells, and other cell which can be a little bit more mature than the initial one. All of those are called spermatogonia. Okay. Now we will see later in the chapter 4 that we have type A and type B spermatogonia, but they are, now we will talk only about spermatogonia. So they will keep on dividing, and here then this cell will divide by mitosis, and those also will divide by mitosis to give other spermatogonia. So we will have many divisions, many mitotic divisions and proliferation, and at certain moment, those spermatogonia, type B, those spermatogonia will increase in size, okay, will enter a growth phase, so will increase in size. And once they increase their size, we will say that they become primary spermatocytes. We will call them primary spermatocytes, okay? So this grow, yeah, so this, after those at certain moment they will stop dividing, they will increase in size. We will say this is the growth phase, and they will become primary spermatocytes. So in this way we generate the primary spermatocytes, which can enter the meiosis to give secondary spermatocytes. As you can see, each cell will divide to give 2 secondary spermatocytes, and each secondary spermatocyte will give 2 spermatids. And in this way, one primary spermatocyte will be at the origin of 4 spermatids. And then the spermatids will mature or will differentiate into spermatozoa. Now, as you can see also that here, when the cells generated from the division of one spermatogonial cell. As you can see, they are connected to each other. Those connections between them are cytoplasmic bridges. Those cytoplasmic bridges are generated because the cytokinesis, after the mitosis here, the cytokinesis is not complete. So the cells, the two total cells, will not get completely separated. And this is the case all over here. In all the mitotic divisions of the cells coming from one spermatogonial cells. They will remain attached to each other by those cytoplasmic bridges. So the cytokinesis is not complete, will not be completed between them, and they will remain attached. Also, the primary spermatocytes generated from those spermatogonia will also be attached to each other. Here also, if we have cells coming from those one, they will be attached to those one, and also the secondary spermatocytes and the spermatids will they are all connected to each other by those cytoplasmic bridges. So what do they serve for, those cytoplasmic bridges? They allow the synchronous development of the cells. So here, whenever they are connected, they can enter the mitosis at the same time. So they will divide, proliferate all of them at the same time. They will undergo the growth phase on this, in the same time. They will, and then the primary spermatocytes, all of them will enter the meiosis at the same time. We will generate the spermatids at the same time. So those bridges, cytoplasmic bridges, allow a synchronous development of the cells descending from one spermatogonial cell. Okay, and why they allow the synchronous development? So why? Because we have those cytoplasmic bridges, we can have a synchronous development. Does anybody know? Because yes.
maybe because السايكل كلها تخلص مع بعضها.
ايه بس انه كيف يعني؟ ليش يعني انه بصير synchronous development لما يكون عندي whole cytoplasmic bridges؟ لانه مثلا حتى تبلش الميتوسيس or الميوسيس او الجروث phase, they need some proteins, some enzymes that will let them engage in this phase. Okay, so whenever they are connected, they can exchange those proteins and enzymes, and so they can all enter the same phase at the same moment. Okay, they will be able to share the same enzymes necessary, let's say, to enter the mitosis, necessary to do the growth phase, necessary to enter the meiosis, etc. Okay.
دكتور، هيدي السynchronization من خلال السيتوبلازميك بريدجز بتصير بالجروث فيز، صح؟
In fact, it is during the growth phase and the maturation phase. The growth phase هي بس لما نقول إنه spermatogonia will become here primary spermatocytes will increase in size and will become the primary spermatocytes. This is the growth phase. Okay. So the during this growth phase, the spermatogonia, and after several mitoses, they will enter a growth phase to become primary spermatocytes. And during the growth phase, so before the growth phase, they were connected to each other, وكان development synchronous alone. During the growth phase كمان حنشوف after the growth phase also. Okay. So whole bridges, they are not limited for the growth phase. And now during the growth phase also, the primary spermatocytes will remain connected through intercellular cytoplasmic bridges which will allow the synchronous differentiation of the enclosed gametes or of the connected gametes. Okay. Now, what we call the maturation phase is the steps where we have the meiosis. So whenever we talk about maturation, we talk about meiosis. Okay, the maturation occurs through meiosis, and the meiosis starts at puberty. We don't have meiosis before puberty. Okay, so the meiosis starts at puberty. So from here, whenever we have primary spermatocytes, as we said, they will engage in meiosis. They will undergo the meiosis 1 to give secondary spermatocytes, and then the secondary spermatocytes will give spermatids, and this is called the maturation the maturation phase. Okay, it is the steps where we have the meiosis. And at the end of the maturation phase, each primary spermatocyte will give rise to 4 spermatids. Okay, each primary spermatocyte will give will give rise to 4 spermatids. Now, the differentiation phase. Now, during all those phases, we still have those cytoplasmic bridges, as you can see. Okay, we still have the cytoplasmic bridges. And during the differentiation, as I have said, we will have cytoplasmic modifications in the spermatids. So they will change their shape. As you can see, the sperm cells, they have a head region and a tail. Okay, and they have this mitochondrial piece in orange. Okay, it is a piece rich in mitochondria, a region rich in mitochondria. We call it the mitochondrial piece. And so they will differentiate to become, to have this shape here. So during the differentiation phase, we will have the spermatids that will be transformed into spermatozoa. And the spermatids will undergo several cytoplasmic modifications to become spermatozoa. They will acquire a tail, a mitochondrial midpiece, and an acrosome. The acrosome is a vesicle rich in enzymes that we will find on the head region, on the upper side of the head region of the sperm cells. Okay. So they will acquire a tail, a mitochondrial piece, and an acrosome, and they will lose a lot of their cytoplasm. Okay, almost all of the spermatid cytoplasm is expelled, will be expelled or lost. Now, the cytoplasmic bridges, the sperm cells will be attached also by cytoplasmic bridges, and when their maturation is reaches its end, when they are formed and they are complete with a head, tail, and a midpiece, those cytoplasmic bridges will be disrupted, and the sperm cells will be released in the lumen of the seminiferous tubule. Okay, so at the end of the differentiation, the cytoplasmic bridges will be disrupted. Here we can see a little bit what happens during the differentiation process. So the differentiation of the spermatids to spermatozoa is called spermiogenesis, and we will see it in details in the chapter 4. So here, as you can see, those are early spermatids. Here the spermatids are becoming more mature or more differentiated. We can see how the shape of the nucleus will become different. The mitochondria in orange, they will migrate to the back side of the cell. They will migrate here. The cytoplasm, a big part of the cytoplasm will be removed or will be lost, okay? And we will have a tail, okay? So this is the differentiation that will give, from a spermatid, we will obtain a sperm cell. Now, here we can review a little bit the different steps of spermatogenesis. At puberty, we have a pool of Spermatogonia that resume their mitotic divisions. So they will divide many times, okay, to give also Spermatogonia, but at a certain moment, those Spermatogonia will stop their mitotic division. They will increase in size. This is the growth phase. So they will increase in size to become primary spermatocytes. And the primary spermatocytes, they will enter the first meiotic division to give secondary spermatocytes, which are haploid cells. The primary spermatocytes were diploid cell, okay? And also the Spermatogonia, they were diploid cells. So now the secondary spermatocytes, they are the result of the first meiotic division. They are haploid cell having 2 chromatids for each chromosome. And now those secondary spermatocytes will have their second meiotic division to give spermatids, which are haploid, and they have 1 chromatid per chromosome. And then those spermatids will differentiate. Here we have the differentiation phase to give the spermatozoa, which are also haploid cells with 1 chromatid per chromosome. Is it clear?
Yes.
Okay. So here also, just 1 minute. During the spermatogenesis, at birth, the testis will contain only spermatogonia or spermatogonial stem cells, which can proliferate. So here we have the proliferation phase, which is during the fetal life, before birth. Okay, at birth, the testis will contain only the spermatogonia. This pool of spermatogonia will be conserved until puberty. At puberty, they will divide again. The mitosis can resume, and we will have a growth phase, a maturation phase, so a growth phase, and the meiosis after puberty, and also the differentiation. So before birth, we only have proliferation and mitotic divisions. After puberty, we will have proliferation. They can always proliferate. So, I mean, during the life of a male, the spermatogonia can proliferate. They can enter a growth phase, can do the meiosis, and to produce spermatids, and the spermatids undergo the differentiation to give the sperm cells. Now, the spermatogenesis occurs in 74 days. It needs 74 days to be completed. And during the spermatogenesis, we have 4 types of cells implicated in this process, which are the spermatogonia, the spermatocytes, the spermatids, and the spermatozoa. So this is all regarding the spermatogenesis. Now I will move to talk about the oogenesis. بس I will stop the recording حتى ما يطلع الفيديو كبير، وبرجع بعمل recording جديد.